Hello, I'm Helen Llewellyn from Anglo-Ruskin University, and this is a quick talk about my undergraduate thesis regarding assessment of positive and negative states in non-human animals. To do this, I used two technologies, infrared thermography, IRT, and facial action coding, FACS. I took left and right lacrimal corankle temperatures from 56 cats. This is the fleshy part of the eye close to the nose, which has previously been used as a proxy for cerebral temperatures. I took this temperature non-invasively using IRT, and I was looking for lateralization, as research suggests that the activation of the right side of the brain indicates negative emotional states. I also used facial action coding, FACS, which codes facial muscle movements and enables us to record them objectively without descriptive ethnograms. For example, you can see here action unit AU201, which is the whisker protractor. I collected FACS data from 122 cats in two treatment groups, a positive event and a negative event. In brief, if an AU was statistically dominant in either group, it was recorded as a positive or negative AU. I then used these findings to code 33 cats with the neurological condition Cerebellar hypoplasia, CH, to determine whether they were performing more positive or negative AUs. This is the PCA, a principal component analysis of my results. A PCA groups variables and shows associations across treatment groups. As you can see by the separation of cats in the negative group, the red circles, from the other groups, there's a clear distinction between negative and positive muscle activations, with CH cats, the squares, aligning more with the positive group, the triangles. The PCA also shows that some facial movements were unlikely to occur concurrently. Some of these were expected, such as the pupil dilator and pupil constrictor. However, the polarization of others is interesting. For example, a half blink is unlikely to happen at the same time as the majority of negative AUs. On this graph, anything pointing right from the centre line denotes the AU was present. The bars pointing left from the centre show an absence of that AU. The length of the line in either direction indicates the significance level. So, by the dominance of the red bars on the right, you can see that I found a wealth of significant muscle activations for the negative event, but none for the positive event. In simple terms, I discovered what an unhappy cat looks like, but not what a happy cat looks like. I find this really interesting, as traditionally animal welfare concentrated on removing negative stimuli rather than increasing positive experiences, and our inability to recognise positive states may confound this further. Also, although CH cats aligned with the positive group, they were more likely to perform negative AUs than their healthy counterparts. The question now is this a welfare concern for CH cats? Finally, only female cats showed a temperature difference during the negative event. The right eye was hotter than the left. And as you can see from the interquartile range represented by the boxes on this graph, this was only weakly significant. The sex differences here could be an artifact of the experiment's design, as male cats may have found the event less stressful. Or it could be physiological, due to sexually dimorphic body temperatures or stress and pain pathways. In conclusion, my research has shown that FACS is a relevant tool for assessing negative states. Further research is required on what happy looks like in cats. That sex disaggregated data is important when discussing animal welfare, and that IRT could be a useful measure for the emotional state of female cats. I would like to say thank you to the Cambridge Cat Clinic and Village Vet for access to their clients' cats for this study, and to Anglo-Ruskin University, the Royal Society of Biology, and the University's Federation for Animal Welfare for recognising my work. Thank you.